Carbohydrates in our diet are present in the form of polysaccharides like starch and also in the form of disaccharides like sucrose and lactose. Digestion of carbohydrates start at mouth where salivary amylase digest alpha 1,4 linkage. Basically, it can break down alpha 1,4 linkage. It cannot break down beta 1,4 linkage. So we cannot digest cellulose. Instead, we can digest starch which contain alpha 1,4 linkage. So when this food reaches at intestine, it break down into disaccharides through pancreatic amylase, and brush borders secrete some disaccharides which further break down these disaccharides into monosaccharides, mean glucose, galactose, and fructose. Glucose and galactose absorbed through a common transporter, which is sodium glucose co-transporter, or we can also call it sodium dependent glucose transporter. It is secondary active transport. Fructose diffuses through glucose transporter 5. So here is a graph. You can see in the graph before the meal our blood glucose level is almost 4 to 5 millimolar, and when we eat in 45 minutes a peak glucose level is achieved which is almost 6 millimolar, and then glucose will absorb, and after almost 90 minutes again same glucose level will be achieved, 5 millimolar, means all the glucose will be absorbed. Like in diabetic patient, this parabola is not found, mean their glucose level remain always high in the blood because this hyperglycemia is caused by deficiency of insulin, either type 1 diabetes or type 2 diabetes. So their glucose absorption is not uniform. So when the glucose is absorbed in the blood, then it absorbed by the body different tissues and different cells. There are 14 different types of glucose transporter. We have mentioned few like glucose transporter 1, which are present on RBCs and brain cells. It has very low km value 1 millimolar, while glucose transporter 3, it is also present in most of the tissues like brain cells, and it also have low km value, and it is responsible for basal uptake of glucose, while glucose 4 is insulin dependent glucose transporter which are present on skeletal muscles and adipose tissues, and it has km value of 5 millimolar, while glucose transporter 2 is present in liver and beta cell of pancreas. It has high km value 15 millimolar, and this 15 millimolar shows that it has low affinity for glucose, while glucose transporter 1 and 3 have high affinity for glucose. Second, we can see that we already discussed that normal glucose level is almost 5 millimolar, while km value of glucose transporter 1 and 3 is 1 millimolar. It means At 5 millimolar at normal glucose level, these glucose transporter provide glucose to brain and RBCs in a constant speed and with its Vmax. When Km is 1 millimolar, mean at 1 millimolar it will take half of its Vmax, and if our blood glucose level is 5 millimolar, then it means it will work at its peak. So when glucose level will be increased, then glucose transporter 2 will work more efficiently and it will accommodate extra amount of glucose. So it has a high Km value, mean it will increase its activity when glucose level will be raised. So it is a very uniform structure that glucose transporter 1 and 3, they are present on brain and RBCs, and they need glucose for their continuous work, and there is a continuous supply of glucose through these transporter. While glucose transporter 2 are present on liver and beta cell of pancreas, which are responsible to manage extra amount of glucose, so they have very high Km value, and God make it very uniform that it will manage extra amount of, mean 3 times the normal amount of glucose. Even liver cell, the glucose transporter which is present on liver cell, it can work with half of its maximum capacity. Here are glucose transporter 4, which are insulin dependent, and most of these transporter are present in the form of vesicle inside the cell in muscle cells, for example. And when insulin is present in response to high level of glucose, it will cause these transporter to fuse with this membrane, and there will be more glucose transporter on the surface of the cell membrane, so it will increase the absorption of glucose. So there is a sudden drop in glucose level is because of this increase in number of glucose transporter 4 on muscle cells and adipose tissues. So when glucose is enter in the cell, it is used by the cell to produce energy. So first step is intrus of glucose, and then first reaction is phosphorylation of glucose, and phosphorylation of glucose is done by hexokinase. Glucose is phosphorylated by hexokinase. Hexokinase mean it is specific for hexose sugar, glucose, fructose or galactose, and kinase mean it transfer phosphate group, it need ATP energy, so it utilize ATP energy and it phosphorylate, or it transfer a phosphate on glucose, and it will convert glucose into glucose 6 phosphate. uh, Basically we are going to get some energy, but for getting some energy we should need to input some energy first. So first process when it glucose enter in the cell is basically breakdown of glucose, and this breakdown of glucose is called glycolysis. And glycolysis have two phases: one is preparatory phase and second is payoff phase. So there are total 10 steps. Five steps are first 5 steps are preparatory phase, in which we input some energy, and we break down it into 2 3 carbon compound, and second phase is payoff phase where we get some energy. So we discuss preparatory phase first, and first reaction is, as we mentioned, phosphorylation of glucose. And why glucose is phosphorylated, or we can say why all uh, intermediates of glycolysis are phosphorylated. So the answer is simple, because if there is a transporter help in transport of for example in this case glucose, and transporter help in transport of glucose along the concentration gradient. So if glucose level is high in blood, it will enter in the cell, and if glucose level is high in the cell, it will move back towards blood. But cell needs to capture this glucose inside the cell, so that whenever it needs some energy, it will utilize this glucose. So to capture this glucose is phosphorylated, because glucose transporters are specific for glucose. They are not specific for glucose 6 phosphate. So glucose 6 phosphate means phosphorylated intermediate cannot pass through transporter. So glucose 6 phosphate is synthesized. Glucose 6 phosphate is converted into fructose 6 phosphate, simple, through an isomerase enzyme. There is no need of energy, and it is a reversible process. Fructose 6 phosphate is converted into fructose 1 6 bisphosphate. So here there is an enzyme phosphofructokinase 1, which basically catalyze this reaction. It is a kinase, it needs ATP energy, and it add another phosphate group at position 1. So fructose 6 phosphate is converted into fructose 1 6 bisphosphate. But this is mean this step is regulatory step, because this phosphofructokinase 1 is allosterically inhibited by high level of ATP or NADH or citrate. Mean when cell is at high energy state and cell is at resting state, mean there is lot of ATP and NADH present in the cell, and cell is at rest, then this high level of ATP or NADH, mean high energy level, will stop phosphorylase 1, and this reaction will stop, mean glycolysis will stop here. If you will perform some function and ATP will be utilized, and then NADH will be utilized to synthesize further ATP, and then all of ATP and NADH utilize, and it will convert into ATP and then ADP and then AMP, and NADH convert into NAD, then it will stimulate again, and phosphorylase work again, and it will start its reaction. So it is highly regulatory step. And if glucose for example glucose level is high and cell is at rest and ATP level will be raised, this step will be stopped, and fructose 6 phosphate will be accumulated, because here is a reversible process. Meaning fructose 6 phosphate will be accumulated and it will convert into glucose 6 phosphate, and glucose 6 phosphate ultimately it stop, it inhibit its own enzyme when it accumulates in large amount, hexokinase. So glucose 6 phosphate inhibit hexokinase, so no further glucose will be phosphorylated, and now extra glucose will be moved back towards blood, and it will enter in the liver cell and beta cell of pancreas. As we mentioned in last slides, there is another glucose transporter, glucose transporter water 2 in beta cell pancreas and insulin and in liver. And when glucose, extra glucose will funnel into the liver, there is another enzyme which is called glucokinase. It is present in the liver cell and beta cell pancreas, and it phosphorylates extra amount of glucose. Same glucokinase has high km value as compared to hexokinase. Hexokinase has high affinity for glucose, but it will be inhibited by extra amount of glucose 6 phosphate by its own it will inhibited by its own product, while glucokinase is regulated by insulin. When glucose level will be high in beta cell pancreas, glucokinase will work, and as a result of glycolysis, more ATP will be produced and more insulin will be released. And when insulin will be released in liver, more glucokinase will be produced, and it will further increase phosphorylation of glucose, and glucose will convert into glucose 6 phosphate, and glucose 6 phosphate convert into fructose 6 phosphate, and fructose 6 phosphate. Basically, there is another enzyme, phosphofructokinase 2, which is not mentioned here, but fructose phosphofructokinase 2 phosphorylate fructose at position 2, and fructose 2 6 bisphosphate in a very small amount. It can trigger phosphofructokinase 1. It enforce phosphofructokinase 1 to start its function, even if uh, its cell is at high energy level. So, phosphofructokinase, even at high energy level, it will produce fructose 1,6 bisphosphate, and then aldolase will break down this fructose 1,6 bisphosphate into three carbon compound, glyceraldehyde 3 phosphate and dihydroxyacetone. So, if a cell is at high energy level, then dihydroxyacetone will be utilized for triglyceride synthesis. Mean dihydroxyacetone will convert into triglycerol 3-phosphate, which act as a backbone for synthesis of triglyceride. But if cell is at low energy state, then glyceraldehyde 3-phosphate will convert it into another compound which is called 1,3-bisphosphoglyceride. 1,3-bisphosphoglycerate is basically 1,3-bisphosphoglycerate can be converted into 2,3-bisphosphoglycerate. I forgot to mention one thing, that the 5 steps which we discussed in last slide, these are basically uh, preparatory phase, and now onward we are discussing payoff phase. Here we get some energy, like glycerol 3 phosphate. When it convert into 1,3-bisphosphoglycerate, it releases a high amount of energy, so that one phosphate is added on position 1, and it will convert into 1,3-bisphosphoglycerate. Secondly, a high energy electron is accepted by NAD, and glyceraldehyde 3-phosphate is converted into 1,3-bisphosphoglycerate, and NADH is produced. Uh, Here we mentioned two NADH. It means one glyceraldehyde 3-phosphate when converted into 1,3-bisphosphoglycerate, one NADH will produce, and then another glyceraldehyde 3-phosphate will undergo same process. So it will create two NADH, and these NADH will enter an electron transport chain, which we will study next, and it will produce an ATP. 1,3-bisphosphoglycerate will convert into 3-phosphoglycerate, and here there is a kinase which is a different kinase, phosphoglycerate kinase, which is a reversible kinase, mean it will synthesize ATP. And in our body, basically there are three steps where ATP is produced at substrate level, when one substrate is converted into a product, and the amount of energy released is utilized by the enzyme to synthesize ATP without electron transport chain. So this is called substrate level of phosphorylation, and here is the first example of substrate level of phosphorylation when 1,3-bisphosphoglycerate is converted into 3-phosphoglycerate. So ATP is produced. So when second 1,3-bisphosphoglycerate will convert into 3-phosphoglycerate, another ATP will produce. So two ATP produced at this step. 3-phosphoglycerate is converted into 2-phosphoglycerate through an enzyme mutase, which is reversible, and 2-phosphoglycerate is converted into phosphoenol pyruvate. Enolization take place. Enolase enzyme catalyze this reaction, and phosphoenol pyruvate is high energy compound which further convert into pyruvate, which is low energy compound. Pyruvate kinase convert this into pyruvate, and it utilize energy to synthesize ATP. So this is second example of substrate level of phosphorylation, and it is also third example of irreversible reaction. First is hexokinase, phosphorylation of glucose, then again phosphorylation of fructose, phosphorylkinase 1, which is irreversible, and last reaction pyruvate kinase, it is also irreversible reaction. So at the end of glycolysis, three uh, uh, three carbon compound pyruvate is two three carbon compound pyruvate synthesized, while 4 ATP and 2 NADH will be synthesized. Sorry. In RBCs, there is some alternative pathway also. 1,3-bisphosphoglycerate is converted into 2,3-bisphosphoglycerate, and then again it will convert into 3-phosphoglycerate. And this 2,3-bisphosphoglycerate has high affinity for beta chain of hemoglobin. It also help in transport of oxygen. Basically, when oxygen bind with hemoglobin, it get associated with iron. So when it is associated with iron, it will not diffuse simply into the tissue. And when this 2,3-bisphosphoglycerate, it binds with, basically it has high affinity for beta chain, so it will bind with beta chain. And when it will bind with beta chain, it will hemoglobin, beta chain of hemoglobin is basically undergo some conformational change. So iron is not available for oxygen, and oxygen simply removed from the hemoglobin. And now, because there is a large amount of oxygen in the RBCs, and in the tissue there is no oxygen, so oxygen is free, so it simply diffuse from RBCs into the tissue. So glycols is not only used for energy purpose, but it also help RBCs to deliver oxygen, to perform its function. Secondly, in RBCs, because there is no nucleus in mature RBCs, and also there is no mitochondria, so NADH synthesized in RBCs, basically they will not utilize an electron transport chain, because electron transport chain take place in inner membrane of mitochondria. So this NADH is utilized in synthesis of or regeneration of hemoglobin, when oxygen get electron from iron, it will convert into ferrous will convert into ferric form, and This NADH provide basically this electron to iron, ferric, which will convert into ferrous. So it helps to regenerate hemoglobin, ferrous, to regenerate met hemoglobin. So glycolysis not only help RBCs to provide energy, but it also help to regenerate hemoglobin to deliver oxygen efficiently, and also it help to provide energy to perform different functions. In other cells, uh, because these NADH will undergo electron transport chains, so one NADH, when it enter an electron transport chain, it will synthesize almost 3 ATP. So if there are 2 NADH, it means there will be 6 NADH, and 4 ATP are synthesized at substrate level of phosphorylation. So 6 plus 4, 10 ATP will be synthesized at end of glycolysis. In other cells, while 2 ATP which are used in preparatory phase, if we minus subtract these 2 ATP, the net gain of ATP at the end of glycolysis is 8 ATP. And in case of RBCs, because NADH is not utilized in electron transport chain, there are 4 ATP synthesized in at substrate level phosphorylation and glycolysis, and 2 NADH, 2 ATP are synthesized. 2 ATP are utilized in preparatory phase, so net gain of ATP in RBCs is 2 ATP. So if you have any question, you can ask.